Hello, my name is Gabriel Carrasco and I will be presenting Epigenetics 101. So epigenetics is the study of changes in organisms caused by modification of gene expression rather than gene alteration of the genetic code itself. Um, these changes can happen by the epigenome having methyl groups or acetyl groups or many of these groups attached to it. Methyl groups makes the genome contract, making DNA unreadable. Acetyl groups makes the genome expand, making the DNA readable. Um, uh, I will show a video showing how these
So this is
changes
the
can
DNA.
happen.
The yarn represents the DNA, which is usually wrapped around the histones, which are these white thingies. The histones have histone tails, which are represented by the blue pipe cleaners, which sometimes attract methyl groups, which makes the DNA contract, making the DNA unrenewable. Once the methyl groups are eliminated, the DNA or the histone tails can then attract acetyl groups, which makes the DNA expand and make it readable. So this is the DNA. The yarn.
So that is how the DNA is affected by methyl and acetyl groups. So why is this important? This is important because changes in the epigenome can affect many illnesses and disorders. For example, cancer, the p53 gene in the body is the main suppressor of tumor suppressor and regulates cell division and cell death. In cancer patients, this is actually methylated. Depression, as you will see later on, will be also affected by epigenetic changes. Epigenetic modifications have a relatively small impact, but they add up. This is a diagram of how epigenetic changes or how cortisol affects epigenetic changes. Cortisol goes into the cell, attaches to glucocorticoid receptors, which then um, these glucocorticoid receptors go into the cell's nucleus and actually activate the FKBP5 gene, which codes a protein that makes the glucocorticoid receptors less effective, reducing stress over time. Mhm. So nutrition is also heavily affected by stress as stress strains the body's resource pool, meaning that poor diet that is filled with deficiencies could potentially affect the creation of neurotransmitters, slow down body cell repair and destruction, disrupt epigenetic processes that may lead to inadvertent epigenetic changes. So medications can also be affected by genes as primarily by affecting the metabolic rate of them. This can affect its effectiveness, that contribute to side effects, and even have non-responsiveness in certain individuals. Um, so there's global differences in gene availability. In Europe, there was a 3% ultra metabolizer, which just means that these people metabolize medications super fast, meaning that in lower doses they could have non-responsiveness, and in higher doses um, they could have like lessened effectiveness, as opposed to Africa, which had 40% of ultra metabolizer genes. Further studies can help personalize drug treatments and make them more effective. So depressive disorders. Um, how do epigenetics changes and genes affect an individual with some sort of depressive disorder? According to our textbooks, the biological perspective on the expression of depressive disorders, low levels of serotonin and norepinephrine are one of the main culprits, with a heightened stress response and immune system response being some of the main causes of our depression. This is supported by the following genes that are associated with depression. As we know, in people with depression, there's usually a serotonin imbalance, which means that somewhere serotonin is being lost. This could be due to an excessive recycling of serotonin, as with SCL6A4. That could be hypomethylated, which means that there isn't enough methylation, or hypermethylated, that meaning that there is too much methylation, making the recycling of serotonin in the brain not as effective or too effective, affecting the serotonin brain balance. The COMT gene could also be affecting it by breaking down too many neurotransmitters or not breaking down enough of them, um, affecting the balance of serotonin in there as well. Um, so schizophrenia is also affected substantially by genetics, as 50% there is a 50% chance of genetical inheritance to offspring of schizophrenia patients. According to the textbook, the expression of this is through gene abnormalities, abnormal dopamine levels, brain circuitry dysfunction. Some of the genes that affect the expression of this could be the DRD2 gene, which encodes a dopamine receptor, which is the receptor that is mainly targeted by the antipsychotic drugs. An insertion deletion of this gene, which just means that there is either an extra copy of it or extra copy of it, or there is actually a part that is missing of it. Um, Affects the response to antipsychotic drugs by either again having too many or too little of the dopamine receptors. Hypo or hypermethylation of this gene could also cause the same effect. The C4 gene is actually pretty important, as it is involved in synaptic pruning, which affects a lot of schizophrenia patients. This could, for example, cause some of the cognitive symptoms of schizophrenia. The microdeletions and duplications in all of these chromosomes are pretty important. However, they are not so well known like what they do. Anxiety disorders. So, according to the textbook, the expression of anxiety disorders is through hyperactive fear circuit, low GABA activity over L improper brain function. Um, the genetics of this, the MAO gene important in the creation of neurotransmitters could be hyper or hypomethylated in this disorder, um, affecting the balance of neurotransmitters in the brain. The NPSRI gene could be affecting it, as the neuropeptide S receptors, or yeah, the neuropeptide S receptors are important in stress response, meaning that they could be hyperactive, or meaning that they would be hyperactive um, in this disorder. The CRHR1 gene then encodes for receptors for corticotrophin releasing hormones. So a hypomethylation of this gene could be creating too many receptors, meaning that there is a lot of corticotrophin release whenever the stress response is activated. The effects on medications are not well studied. However, some of the some of the previous medications, such as SSRI, that is affected by the metabolic rate of each person, could since SSRIs could also be used for anxiety disorder, the same kind of genetics could apply to this as well. So the key takeaways are that epigenetic changes can increase or decrease the risk of psychological disorders. The risk increases, however, are quite minute per gene, but they do add up. More research is needed to obtain causal relationship of genes, and epigenetic changes are reversible. So it's not like you're locked in if you have an epigenetic change. These are my references. Thank you for listening.